Hello, thanks for your interest in natural product discovery. Today we're going to explore what factors make for a good natural product to be discovered. I mean, if you want a powerful drug, something that has a powerful effect on bodies, could be a human, it could be a pet. Sometimes there's more money into the pets industry than the humans. Then you have to look for certain characteristics of the product. And I'm going to summarize them in only four. So if you're interested on which four factors make for a good natural product to be developed into a drug, be my guest for the next minutes and enjoy this small taster of our natural products discovery master at Liverpool Moors. As I said, critical factors that make for a good drug to be developed or good natural product to be developed into a drug are first of all the drugability. I mean, this natural product can be made into a drug. Drug needs to have very selective activity on a given receptor. So the mechanism of action is clear for us, so we can harness both its efficacy but also prevent any toxicities because we know what it's doing exactly in the body and where. That's quite difficult sometimes. Of course, it wouldn't be a good drug if for all the potency and selectivity, it doesn't get into your body and do the work we want it to do. Well, that's what we call bioavailability. Well, what routes of administrations this molecule will need? Is there any transporters issues within the body? I mean, is the body able to transport the drug inside to the right tissue? Is it the drug metabolized before it's doing anything at all? That's a very important factor. It looks a very difficult one, and it's actually very complicated to find out in the lab what all these issues. But there's a very clever rule that is almost mathematical, and we are going to use it to just kickstart our drug discovery process, saying yes or no to these possible issues on bioavailability. Of course, a molecule, a natural product is a molecule. A molecule is a single chemical entity. We need to know the chemical aspects around this nice natural product. If it is not stable, it's of no use. If it is from a very rare plant, it's not sustainable to collect the plants and then extract them. We may have to go to the lab and create the molecule from scratch in the lab. Or the molecule might be very promising, but still we need to squeeze a little bit more potency out of it. Then we have to make semi-synthetic derivatives that are maybe more efficacious, but also more bioavailable. And sorry, I'm hiding this. The business side needs for the sources to be sustainable. So the sourcing of the drug have to be sustainable in time. We don't run out of it. But also patentability. Can we patent it? We need money. We work for something that is called money. I mean, we want the greater good for humankind, but we need to give something back to all the industrial partners that are making possible this discovery and marketing. Well, let's start with comparing natural drugs, natural products with more synthetic products. The problem is that natural products, mostly as they are found in nature, they defy common understanding and common rules of the game that we were talking about before. So look at the critical factors according to the books, and then we are going to see how natural products are mostly against the book. As I said, we need a specificity on our receptor activity and selectivity. We need the drug to be good in order to not to be metabolized too quickly and arrive to the place of action, to the site of action easily. Also has to have physicochemical characteristics that make it easy to work with and easy to enter and arrive to the place in the body you want. For this, we are going to use the Lipinski rules of 5. That's quite a genius thing to do. Of course, this molecule have to be stable. Maybe we need it to be easily synthesized. We'll see. And obviously, we want also cash on it, patentability, and hopefully not sharing too much benefits with others. I will tell you what is this meaning. Natural products problem is that many of them are multi-target. They act on many receptors. They've been made for plants as dirty molecules. What is a dirty molecule? We'll go into in deep a little bit later. But these are molecules that do many things. And that's why natural products are really fantastic. For example, curcumin. There's a craze about curcumin from turmeric, the orange curry ingredient. Curcumin is the molecule that makes for this bright color, but also it's an antioxidant, anti-cancer, anti-inflammatory, you name it. It's doing everything. It's a dirty molecule. It's wonderful, but it's so wonderful that it's of almost no use because it takes too many targets in the body. So we have maybe a molecule that is trying to do too many things and not specifically something. It might be a blessing in disguise or it might be a no-go. Well, the future will say. It's difficult to ascertain if an extract, natural products start as extracts of plants, and sometimes the extracts are very powerful. When you divide the extract into their different components, they lose the activity. But as extract, they work very well. It's difficult to forecast if the metabolism, the metabolism and transporters issues for a myriad of compounds, what will be? It's almost impossible. Also, for extracts, it's really difficult to ascertain the overall physicochemical issues that we may face for their many components. Most of natural products defy the famous Lipinski rule. We will see an example later. Mainly because they are impossibly big, sometimes poor water solubility, and extraordinary stereochemistry, and that makes actually the case for many natural products to go into the market because they're so impossibly stereochemical that do impossible things that are reserved only for kitchen's nature actually. Many are impossible to synthesize, as I said before, very complex chemistry. They have to be isolated from the nature and maybe just slightly modified and unpacked for the people. But obviously, if something is natural, cannot be synthesized, sorry, cannot be patented. You cannot patent sugar. Someone very clever trying to patent sugar and then we all have to pay him for every time we have a sugary coffee or add sugar to a cake. So we cannot patent DNA, we cannot patent anything that is present in nature. It belongs to mankind. We can maybe patent several applications or several ways to extract it. We'll talk afterwards. But this is again a problem that hinders natural products discovery going into the big market. And obviously, many natural products we find them because there's ancestral knowledge. There's this old medicine man who says this natural product works, or your money back. Well, then if we depart from this information, it's all fair that we have to give back to the medicine man or the culture who told us this have to be used for this purpose. This is called benefit sharing, and we'll see a little bit more afterwards. Let's make an example of a, a very good drug, which is natural almost, yeah, which is natural. Let's say salicylic acid. Salicylic acid from willow bark, from the willow tree, the bark of the willow tree, makes the precursor for aspirin. Aspirin is just slightly modified salicin, so it's not too acidic to our stomachs. This is salicin, that is almost like you say, well, it's present in the willow bark tree, and is we have here the precursor for salicylic acid, and this is a sugar. Many times the plants store powerful molecules like salicin in the form of sugars. This is kind of a very clever strategy to make them not very poisonous or toxic, or just deactivate them in waiting to be activated when the insects or the animals try to eat the plant. So the sugar is cleaved in our mouth with the saliva enzymes, and then it releases the real thing. So the salicylic acid is too acidic because we have a phenol. Phenols are very acidic, plus another acid, which is a carboxylic acid. So a clever German chemist decided to make an ester in the phenol, and this makes for a less acidic molecule that we can stomach literally. Otherwise, and still, if we abuse or we use chronically aspirin for a long time at high doses, we may develop an ulcer, a hole in your stomach. Right, so wonderful. Salicylic acid is a natural product that fits all the rules that we said before for a wonderful drug, a very good one. So it's soluble in water, but also in alcohol and many other solvents, organic solvents. It's quite a good molecule that it likes any way to soluble. It's soluble in many ways. It's a small and simple. We don't have stereochemistry. So it's almost ready to be synthesized by ourselves. We want from scratch or semi-synthesized easily. Has no chiral center, so it doesn't make the synthesis too complicated. Therefore, nowadays aspirin is not extracted anymore. It's a long time that it's not extracted anymore from willow bark. It's just made in a lab from scratch. But if you want it, if you wanted it, you could easily source salicylic acid from nature, because just 10 grams of the Salix alba willow tree bark, 10 grams of dry bark contains 1 gram of salicylic acid. So it's an enormous amount of it. So the rest are fibers and stuff in the bark. And beautifully, it's a selective COX-1 inhibitor, has a selective a mechanism of action, which is the very first critical factor I was talking about to you. So that's a deal of a molecule which is natural to be made into a successful drug. Actually, it was so successful that the Germans were cashing with aspirin millions and millions and millions. So much that after World War II, the Allied forces in one of the paragraphs of the surrender of Germany contained that they had to give up the patent for aspirin. So in order to repay, and that would repay for the cost of the Allied forces to the war efforts. That was so big, the market of aspirin at the time. So, but yeah, it means that aspirin could be patented. It's a molecule that can be patented because it's not present in nature anymore. You make an ester out of their natural product. So it's your molecule now. It's a new molecule that doesn't exist really in nature. It comes from, or it came from, but not anymore. And yes, willow tree bark was known since ancient times as an anti-inflammatory infusion, but it's so ancient the knowledge and so widespread that you couldn't find anyone to to share the benefits of aspirin with. It belonged to humankind in a way. So the discovery of aspirin doesn't wouldn't qualify probably for benefit sharing nowadays because it's being reported since thousands, not thousands, but hundreds of years its use. So we are lost track who we had to share the benefits with. This beauty of a molecule is much bigger than aspirin is Taxol or paclitaxel. It's from the another bark of a tree. This time it's the yew tree. Right. This is indicated as an oncological drug for ovarian, breast, and lung and pancreatic cancers. It's a very big molecule, you'll see, and that's a natural one, and this will bring some problems now. First of all, their discoverers, Wall and Woney, said that it was as soluble as brick powder. I mean, it's almost impossible to solubilize in anything. They had to invent a special formulation to solubilize Taxol. It's so big and complex structure with so many chiral centers. We can synthesize it, but it's so difficult that it's better to produce it from natural organisms. If you come to the master, we will explain you how there's not anymore the tree, the source, but some endophytic fungi. On top of this is extremely rare in nature. 1 gram of purified Taxol would need about 10 kilograms of plant bark, dry plant bark, and this is equivalent to a skin out, to skin 4 to 5 trees. And when you skin a tree out, you take out the bark. It's like skinning an animal. It's horrible. The tree will die, and is not sustainable really. The good thing is that we found Taxol to be first in class, first in kind of drug. For first time, a molecule that could freeze tubulin. So any cell that was in need to divide, and cancer cells are in need to divide all the time, it freezes in the middle of the division, and thanks due to Taxol. So this was really the deal breaker for Taxol to become a very successful drug that is so selective, and that's why this factor I've put it at the beginning is so selective, the mechanism of action, that is really a deal breaker. It has to be offered to the patients. Obviously, you cannot patent Taxol as you see it here. Yeah, you see it there, up there. Is as it is in nature. So you cannot patent Taxol. Nobody can. You could patent how to obtain it by certain chemical processes, or how to apply to different cancers. If you run the clinical trials, they cost a lot of money. You can patent the outcome of the clinical trial, fair enough. And there wasn't any benefits sharing here. There wasn't any medicine man telling us GU bark is good for cancers. It was just found randomly in a big quest for anti-cancer drugs from nature. So in this case, it's a case of natural product that doesn't come from ancient or traditional knowledge, and we don't have to share the benefits of this knowledge with anyone. So this was offsetting the red no-goes in critical factors. So then again, looking at Taxol, you see that although ideally I told you there are some critical factors that a successful drug should tick all the boxes in, we can live without some of them as long as the natural product provides such an edge to the application that offsets all the other upsets. Right, let's start by the mechanism of action, activity and selectivity. I said this is the 4 key. This is why, for example, molecules like Taxol are developed into drugs, even if you cannot really synthesize, if you cannot patent it, you cannot, it's difficult to obtain, but well, it's worth the efforts and the trouble because it's so active and so selective. And nothing else in nature does it before or out of a lab was able to do it. So it was to discover it and to bring it into a drug. As I said, many natural products are not that selective. Taxol and aspirin are very selective. There's a bull's eye on a target. But most natural products like curcumin from turmeric that I was commenting, it's acting on so many targets and that there's too much noise in their mechanism for actions. So in the terminology of drug discoverers, we call Taxol and aspirin clean drugs. They are very, very precise in their target, and turmeric and curcumin would be a dirty drug. They're acting on so many places. It's important to say that clean or dirty drugs do not denote safety or efficacy. As I said, curcumin could be one of the best anti-inflammatory drugs around if we can harness its potential somehow. But it's, as I said, very dirty molecule. It does many other things at the same time. So clean drug, again, is something that is biochemically very, very much hitting only one target. So the mechanism of action is known. This makes for a rational drug development. So these molecules preferably bind to a single target, have no side effects that we don't know, or at least they have side effects that we will anticipate. Samples, Taxol, aspirin, as I said, but the dirty drugs are those that the mechanism of action are unclear. Therefore, it's hard to know whether you got some specificity on anything or how it's working. And yeah, it could be good. Medicine started with many dirty drugs. Origins of modern medicine started with many, many dirty drugs that were efficacious, but they had very many confounding aspects and some toxicities probably. For example, one interesting drug nowadays that we use a lot for anti-diabetic activity is metformin, which is derived, like aspirin, is derived from salicylic acid from the willow bark. Metformin is kind of semi-synthesized natural product that is present in Gallego officinalis or French lilac or goat rue. and was used for medicine for several centuries. Well, next thing is, okay, if we have a clean molecule, will it arrive to the place we want? Will it do their job and 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 go over all the hurdles that our living organisms puts to molecules? We have barriers, we have membranes, we have tissues, we have livers that destroy poison. So will it make it to the place? Very difficult question to answer. But this guy, this happy chap here, Chris Lipinski, made a very easy, or came out with a very easy rule of 5. It's not because it's 5 rules, it's only 4, but they are around the number 5. That makes a little bit easy to forecast, to predict if the molecule is going to be more or less good in terms of bioavailability or not. So for example, if we look at Taxol and aspirin again, the rules of 5, which is a physicochemical approach to predict bioavailability, would ask you very simply first, what the molecular mass of the molecule is. If it is less than 500 Dalton, then it would be a good one. Taxol says no, I'm very chunky molecule, very tubby and big, so I'm waiting far off this scale of 500 Daltons. As much as 853 is a no-go according to the Lipinski rule. Aspirin is light and nice, so yes. Then second Lipinski rule is the molecule has to have no more than 5 hydrogen bond donors, and these bond donors are NHs, amines, or hydroxyl. So it's about going around and finding how many of them do we have. Another one here, another one here. So we have 1, 2, 3, 4. Yes, it complies Lipinski rule, Taxol. It's very big, but it only have 4 and no more than 5, certainly, NHs and OHs. And what about aspirin? Well, aspirin only has this one, this hydroxyl, but it has no amine, only one, so even less than those. Yes, but it's good. Both are good in this part of the Lipinski rules. Also, Lipinski, Chris said no more than 10 hydrogen bond acceptors. So no. So these are all nitrogens and all oxygens put together. So if you count or you go to the formula, the chemical formula of Taxol, you will find that Taxol has a combined 15 atoms of nitrogen and oxygen. But aspirin is only 1, 2, 3, 4 oxygen. So yeah, it complies the Lipinski rule. So for now, we have no, and yes, for aspirin. Another thing that Lipinski said is very important is what they call the partition coefficient or log of P. You will have more information if you come to our master. Logarithm of P is the solubility in between water and non-miscible with water non-polar solvents, usually octanol. Taxol, unfortunately, has a very non-polar behavior, or overall characteristic is non-polar. So doesn't comply with the Lipinski rule because it goes well beyond the 5, it's 7.3. Whereas aspirin is close to 0 and it's a yes. Yeah. So after all, Lipinski rules hold quite true for us as as potential for being a drug. Um, Aspirin is much better. It's zero violations of Lipinski rule. Taxol violates Lipinski rules many times, 3 times. Doesn't mean that Taxol has been a very successful molecule and is very good, but it means, according to Lipinski rule, that we will have a lot of problems administering Taxol. And in fact, it has to be administered within a very specific formulation, and it's difficult for Taxol to arrive to the places they need, and it have to be injected in your veins directly. It cannot be taken orally just yet, at least. So it's quite difficult to get it to the place it have to work. Obviously, apart from all these drug metabolism issues, also include the liver. The liver is a wonderful organ, and we only have one of them. So it's destroying everything that is not a nutrient, anything that is not a sugar, fat, or vitamin, etc., is destroyed by the liver. So your natural product will be destroyed by the liver mostly if it is not a sugar or a fat or etc. So metabolism will diminish the efficacy of the drug or the medicine, but sometimes there are some natural products that need the liver to be activated, and then it becomes we call them pro-drugs. They are administered in the inactive form, and they are metabolized and then become active. We have to know how it works in this sense, because it's an important concept that we can play with, and actually pro-drugs are very, very good candidates for medicines. Well, as natural products go, they're chemical entities, complex or simple. But we want simple chemical entities, just single molecules. We have to look at the stability of the total synthesis, and if we can make derivatives out of it, because all of them are important for a successful natural product to become a molecule, a drug. The stability of a drug can be, well, in face of many challenges that are physical, chemical, microbiological, biological, and therapeutic. So the stability of a drug, for example, physically speaking, is that if the drug is going to precipitate, to get absorbed somewhere, if it is going to change the color, etc. Chemically speaking, we cannot get a medicine that is losing activity over the time, just because it degrades by itself. By itself, I mean in contact with oxygen, for example. Also, if the drug is attacked and digested by microbes. Microbes are everywhere, even in medicines. So we have to look at how the drug is able to survive the microbe attack. Biologically speaking, if they are unstable, they may be toxic, so we don't want it. And therapeutically speaking, the medicine, if it is not stable, the drug might be not toxic, but it eventually degrades and makes the medicine no effective anymore. From my point of view of a chemist, I'd like you to always think about these 4 very important challenges for any chemical entity, and particularly natural products. UV radiations, I mean, is it stable under sunlight? For example, hyperisine from St. John's wort. reacts with light and produces radicals and then makes phototoxicity reaction. Hydrolysis, water is everywhere, even in the air. Otherwise, we wouldn't be alive. So we have to look at if the molecule is stable in contact with water, or even soluble in water is stable long term, or when it's soluble in water at which pH is stable and with pH is not stable. For example, ivermectins that are #1 antiparasitic drugs and are natural from natural products. They're natural, yeah. Very stable to heat, for example, to UV radiations too, but not to basic pH. So, but they can be very happily living in very acidic water. We have to know beforehand. Oxidation, obviously, is stable in open air, is stable in our Earth atmosphere, which is rich in oxygen. For example, unsaturated fatty acids such as omega-3 would become rancid over the time if we don't protect them from the oxygen. And also, when the natural products get into a cell or into an organism, they may be degraded by extra or intracellular enzymes. And one of the most ubiquitous are esterases. For example, aspirin, which is an ester of salicylic acid, could and is very easily hydrolyzed into salicylic acid again inside many cells in the body. When we have wonderful natural products like artemisinin, which is the anti-malarial #1 in the world right now, this is the natural thing. This is the natural product as it is happening in the plant and as we can obtain it from the plant. But we discovered that all these semi-synthetic derivatives are not only more bioavailable, they can enter in our body more easily, but also more potently, acting on the parasite. For example, artesunate is all about, all these derivatives are all about making a bit of change in this ketone, in this lactone ring. So, for example, if you add all this, you made artesunate, or if you just a methyl only, you have artemether, you can patent it. You can patent it because now they are not anymore natural. Or this one is M934, okay. Or you just change the ketone for a hydroxyl. You add some hydrogens there and take out the double bond. All these semi-synthetic derivatives are interesting because you can patent them and cash them, but also they used to be more bioavailable. They enter your body because these extensions make them more lipophilic and they can cross membranes and then they can enter the body better. And then in the body, this can be hydrolyzed because we have esterases, and then we have the artemisinin free again inside our organism. But well, if you want to go the hard way, you can create artemisinin on your lab from scratch, from strange molecules and with a lot of, well, not a lot, but it's quite short as a synthesis. In 7 steps, they say. Well, probably we have to synthesize first this, obviously. But yeah, and then it's not easy to synthesize. We have yields that are not very high. In the end, it's 15% of what you get in going out as artemisinin. But well, you could do it if you have no access to the plant or you want to not use the plant. You could create artemisinin in your lab. So it's important that this is possible, even if remotely, because if we run out of the natural source, we can reproduce it in our labs. So the business aspect, as I said, have is the not the last, but probably the first thing you have to think about a natural product. If I want this natural product to be a medicine, will I have to share my benefits with someone? Ask permissions? Can I make it my own one by patentability, etc.? So let's have a look at it. Sustainability is a very complex thing. As I said, it depends on many factors. How rare is the source of it, or how abundant is the source? If maybe very abundant, but the source is in the deep ocean, so it's very difficult to get it, etc., etc., and all the messing you do around in nature to get it from nature. So it's a very complex thing that depends on many parameters, and therefore there are some impact indexes. I'll show you one later that measure all these parameters and tell the pharma industry how sustainable will be this molecule to go by. Obviously, harvesting, as I said, the natural source could be rare or could be abundant, but in a very difficult place to arrive. So that's paramount to take into account. Once we get it in our labs, or if we want to synthesize it, maybe it's not sustainable because we have to use very, very toxic processes, and then it's a bad idea. I mean, we can live with it if the medicine is going for the greater good, but if we can do it the green way, all the better. Dr. Fayez Ismail in our master will teach you what steps do you have to take towards green chemistry. If you want to go into this extraordinary and complex world of sustainability and herbal medicines and pharma products, go to this fantastic paper on ecopharmacognosy, natural products research in phytochemistry letters. This is one of the many indexes that the pharma industry or the natural products discovery industry is using, the Green Impact Index. And if you click here, for example, you have a kind of vision of how it is applied and how it works. So to measure the sociological impacts and as well as environmental impacts of the production or sourcing natural products for as medicines. As I said at the beginning, we want to get some money out of it. That's why we are studying many years in our life, and also that's the only way we can protect the market and get many natural products into wonderful medicines. We like it or not, but nobody's going to do it for no money. So patentability is very difficult, as I said, for natural products. A natural product as it is in nature cannot be patented. Think of sugar, it cannot be just patented by myself, for example, and I make all of you to pay for sugar from now on. It's in DNA. DNA, well, you cannot patent it. The DNA that is inside a person, that is inside any living organism, belongs to all the mankind. So what can we patent in terms of natural products? Look, in the United States, they talk about significant differences. It's in the United States, they say that if a natural product is not patentable, actually, but if you extract it from nature and process it in a way that at the very end the medicine doesn't resemble anymore what is in nature. Even if it is from nature and we don't change it chemically, we think about a very refined extract, only a subset of the chemicals that are present in the plant, then you can do it. You can patent the process of it, and you can patent the final product for you, right? In our master, we are explaining this in terms of the case study of dragon's blood from the rainforest going into being Mytesi, which is a wonderful drug for AIDS patients. It's cutting down on non-infectious diarrheas they have. Also, the European Union says that in the same way as the United States, that if you isolate from this molecule from natural environment and the process is quite complex, you can patent this process, and in the end the final product is nothing that you can readily see in nature. So you can patent this extraction and this isolation of the process of the product. The product itself will always belong to mankind, but the way to get it will be yours. Okay. This is also the technical effect. It's called the technical effect. So how I go about getting the final product, I can patent it. I think these three things in the end talk about the same. Also about the includes the last one includes the use of it. Maybe you cannot patent Taxol, but you can patent its effect on breast cancer if you do the clinical trials and you pay for them. And as I said, but last but not least, if you are discovering things because someone told you, it would be very, very rude not to share your benefits with the friend who told you you have to look there. There's a good natural product in there. So that's what we call it benefit sharing. And it's also quite complicated. We have now what we call the Nagoya Protocol and the Biodiversity Convention, you may have heard of. There's a lot of things that you have to consider, but basically, if you depart on your natural products discovery from someone hinting to you where the source is, you have to share your benefits with them. Also, if you take your product from a plant that only grows in one country, this country is the owner of the plant, and you have to pay this country repay some benefits to this country. So if you liked this analysis of the critical factors, well, it's a good thing because then you could become a good natural products discoverer. Actually, we are using this drug camptothecin from Canthoteca acuminata. It's a tree. It's a rainforest tree, originally from China, that yields this wonderful molecule that is anti-cancer also. And what we do in class is reading the books and the papers about camptothecin and extracting all the information that help us to analyze whether camptothecin meets all these critical factors or not. And then we discuss whether it would be a good business or not to get it as a drug lead. So this is the way we teach in our master. It's very much case studies. There's no any more exams that we do just knowing by heart everything. You always have all the books in the world to look and research and make decisions. But obviously you need to understand all these critical factors inside out. What I just mentioned is just the tip of the iceberg. These critical factors in deep have a lot of to say for each one. and there are many great songs that we discuss in the lecture room. It's great fun to have these workshops and give thumbs up or thumbs down to a molecule depending on how it fares on all these critical factors. Well, thank you very much. And then again, if you liked this and you are interested in our master, subscribe, contact us at Liverpool Young Moors, and we can discuss with you whether you would be a good candidate for our program. Until then, thanks for watching, subscribe, and see you later.